Hello everyone, welcome. We have already talked about immune system, how we have the primary lymphoid system and we have the secondary lymphoid system. Talked about what is immunity. Now we are going to describe immunity. Immunity is it's the response against the antigen, how it stays if or any foreign antigen stuck. Okay, so how it fights against various antigens. So that is the immunity, its ability of the immune system to fight against disease-causing organism. Now, if I talk about types of immunity, I have an innate immunity, which the immunity that I have got inborn, and I have got an acquired immunity, which is adaptive. So I acquire it with my lifetime. Okay. So if I talk about the immunity that is there, if I talk about innate immunity, if I talk about innate immunity, okay, this is a non-specific difference present at the time of birth. So there are four types of barriers. It is the barriers it needs to, every antigen it needs to enter after crossing various barriers. It's like the doors. It will have to knock the door, the permission is given, and then it enters into. Okay. So there are four barriers. I have a physical barrier, like skin is the barrier, physiological barriers are there, cellular barriers, the cytokine barriers. So there are different barriers that I have. Okay. Now, if I talk about the physical barrier, this is the first line of defense. Like maybe saliva, the tears, skin, mucus linings, gastric hydrochloride, good gut bacteria, all this. So skin is providing the first line of defense wherein it can stop the foreign antigens to enter into the body. The mucus coating of the lining in the respiratory, gastrointestinal, and urogenital tract to trap the microbes. So, saliva, it has antibacterial enzymes. Tears, again, antibacterial enzymes. So if something enters into your eyes, the tears will come out and it will move off. Skin prevents the entry of pathogen. Mucus lining, it traps dirts and microbes. Gastric hydrochloride, it the low pH, it will kill the pathogens. And good gut bacteria, one compete the bad bacteria which is there. Okay, so this is the first line of defense that I have. Next, I have the physiological barriers. Physiological barriers would mean the gastric, HCl, saliva, tears, all these are physiological barriers. Skin is the physical barrier, but what is coming out like saliva or say tears, all these are the physiological barriers that we have. So they prevent the microbial growth. Next, we are coming into the cellular barriers. Cellular barriers are like white blood cells. White blood cells, what is the main function of white blood cell? It is to fight against diseases. We know it from very small age. So they are phagocytes like WBCs, polymorphonuclear leukocytes, that's PMNL, monocytes, and NK lymphocytes, that's natural killer cells, macrophages, etc., which is there, which is going to fight against various diseases. So we have neutrophils, we have monocytes, natural killer cells, or macrophages, which is going to fight against the antigens which has entered. Next, we have cytokine barriers. The virus infected cells secretes a cytokine proteins which is known as interferon. So if say the virus is infected, so skin did not stop. We have physiological barriers like tears or saliva, all this did not stop. We could have had macrophages, it could have stopped entering into the cells. We had WBC cells, yet that could not serve. It has entered into the cell. Once it has entered into the cell, what will happen? The cells will secrete a protein called as interferon, and it will protect the non-infected cells from further viral infection. Okay, so that is what is going to happen. All right, next we are coming from to acquired. So now we have completed the natural immunity or innate immunity. Now we are coming to acquired immunity or passive immunity. Now, acquired immunity is what? Pathogen-specific immunity developed during lifetime. It is characterized by memory, that is the first encounter of a pathogen body produces first response or the primary response in low intensity. Second time if it occurs, the intensity becomes higher. So first response, you have a very less immunity, and in the second instance, you have a build immunity that is going to happen. So in the first time, there is a production of the B cells. The second time when it enters, it will say, I remember this one. Red alert, red alert, red alert. So memory cells will give a red alert to the antigen, which is there. Okay. Next. Now the primary and the secondary immune responses are carried out by B cells and the T cells. Okay, T lymphocytes. So B lymphocytes, they produce antibodies. These are proteins to fight with pathogen. And T lymphocytes, it will help the B lymphocytes produce the antibodies. So how is it occurring? The B lymphocytes are there. So if the B cells, the B receptors are there. So B cells, the antigen will come, it will bind. Once it binds, there is a chemical signal that is produced by T helper cells. This T helper cells, in turn, what will do? It becomes a plasma cell. So this plasma cells will produce number of antibodies. So number of antibodies. So this is how various my immune system will occur. Now in the acquired immune system, I have got immunoglobulins or the antibody. Now there are various types of antibodies: IgA, IgG, IgM, IgE, and IgD. So these antibodies, they contain 2 light chains and 2 heavy chains. So if you see that these are the heavy chains, so when I have this is a heavy chain of the antibody that I have. So 2 heavy chains I have, and then I have 2 light chains. So these are the 2 light chains. So this, if you see, these are the 2 light chains and these are the 2 heavy chains. So now once I have the heavy chain and the light chain, so they are being connected with disulfide bridges. And towards the end, these area are antigen binding sites that I have. This area is the antigen binding site. So if I have to draw an antigen binding site, I can always draw in this way so that it will show me a variability, but it should be complementing the site. So antigen variable site that is there. Okay. So now if I say IgG, immunoglobulin G, which is there, IgE, okay, again a monomer, IgE, IgD. IgA is a dimer because it has a two immunoglobulins which is attached by a joining chain. Pentamer is IgM. IgM is a pentamer. That is there are 5 immunoglobulins which is put together in the joining chain causing a pentamer, that is IgM. All right. Now, if I have, there are 2 types of acquired immune response. One is humoral or antibody mediated response or antibody mediated immunity, AMI, and other is cell mediated immunity. Now, if I talk about humoral and antibody mediated immunity or AMI, I will say, so there is an antibody coat. So there's a virus which is coated with antibodies, which is there. These antibodies will go, there is the epitope or antibody, it is the attached site is the epitope is there. Okay. Now the virus, this virus, they cannot infect the cell. So they are disarmed or they cannot infect the cell. So antibodies, they are found in the plasma. So now this viruses, they are permitted. So uninfected host cells are there. Now this tagged virus is destroyed. Once this virus is destroyed, then there are antibodies which has got created in the plasma, blood plasma. So they are known as humoral immune response. Next, I have acquired immunity. That is the next type. Okay, in the same type. So here in cell-mediated response, which is their CMI, cell-mediated immunity. So in this, what happens? The immune system response is mediated by T cell. So earlier I was talking about B cell. Now it is a T cell. So the T cells makes contact with the release granules. So I have the virus infected host cell. So this virus infected host cell, it will get contacted with cytotoxic T cell. These T cells, these granules, it will induce cells to self-destruct the virus, which cannot replicate. So the body can differentiate self and non-self. And the CMI, it causes graft rejection. So there is a graft. Graft means there might be a skin graft or some other organs would have been grafted into yours. It will say no. Now, if I talk about tissue mismatching or blood group matching. So tissue matching or blood group matching. So there might be a donor and the donor would have given a tissue to the patient. So in case if there is, this is called as a transplant. Okay. So it is very important that I see that tissues are going to match. Or, the blood, when I transferred the blood, I would have heard, okay, fine, this blood group I cannot give. A blood group, I cannot give to a B blood group. So I have got these cases. So why? So that there is a mismatch. Otherwise, there would be a rejection which will take place. And it will just consider the foreign tissue to be non-self, and it will kill the tissue, even if it is required by me. But after all, it is a foreign, if I have got a heart transplant. So it is a foreign tissue, right? So my body will start rejecting the heart. Okay? So even the blood transfusion, the same will occur. So O can give to O. So, O is a universal donor. So O can give to O, A, B, anything. Whereas if a person with A can give to A and AB alone, cannot give to O, cannot give to B. B can give to B and A, whereas AB can give blood only to A. So you should know who can be a recipient and who can be a donor for that purpose. Now, this acquired immunity that I'm going to acquire can be active, can be passive. So acquired immunity, which is active, is natural or artificial. When I say natural, I have been infected by the virus and I'm now having an immunity. Say chickenpox. Once I'm infected with chickenpox, it's stated that you cannot get it the second time. That is very common. So I'm being infected and I am having an immunity to the disease. Whereas artificial would be if it is by a vaccine. I am vaccinated against polio. Hence, I might be, there is a very high chance I would not get a polio. Again, passive also, it might be natural or artificial. Natural immunity is the maternal antibodies which gets transformed to me via the maternal chromosomes or while, when the child is being fed in the early days of the life. Okay, so a huge immunity is from the mother's milk, and artificial is by taking up some monoclonal antibodies which is there. So, if I'm talking about active immunity, it is the immunity in which antibodies are produced in a host body when the host is exposed to antigens. Okay, example, living or dead microbes or other proteins. And this is a very, very slow process. And it is produced in two ways. Natural active immunity, it is developed during natural infections by the microbes. I told you, it is I am being infected and then I am getting antibodies to the disease. Whereas if I'm talking about artificial active immunity, I'm talking it is developed by injecting microbes deliberately into during immunization. So immunizing the child by some kind of vaccine. Okay. Now passive immunity on the other side, if I say natural passive immunity, whenever I'm talking about passive immunity, I'm injecting ready-made antibodies. So natural passive immunity would mean antibodies like IgG from mothers, that is placenta to the fetus, antibodies like IgA in colostrum infants, that is being there. So that is a natural passive immunity, whereas artificial passive immunity, anti-tetanus syrup. So most of us take tetanus, right? If we get bit by, if we harm ourselves or hurt ourselves, we take tetanus. So that is an artificial passive immunity. So antibodies are there. I'm injecting the antibody directly into the body. So if I'm talking about immunization, when I'm trying to immunize a child, it is based on the memory of the immune system. It can be active immunization or passive immunization. So active immunization is basically in the child when you take the polio vaccines and all those. Passive immunization, we talked about active immunity, which is right, and passive is the antibodies are being injected. Kill that virus, if it is injected, it is active immunization. So polio or something is active immunization. Okay, antibodies produced within the body. Whereas passive immunization is antibody is injected into the body, ready-made antibody. So tetanus is a passive immunization. So there are two kinds of immunization. Okay, acquired immunization, a preparation of vaccine, as I was telling you, is a preparation of vaccine which is introduced into the body. It is result in development of antibodies which is inside the body. Inside the child's body, the antibodies are being produced. During actual infection, the antibodies neutralize the antigen. Okay, this is active immunization. And in the passive immunization, what is happening? In the passive immunization, okay, before going to passive immunization, active immunization, the vaccines also generate memory B and T cells. They recognize the pathogen quickly, like polio vaccine, hepatitis B, DPT vaccines, etc. Vaccines are produced using DNA recombinant technology. And if I talk about passive immunization, then it is direct injection of pre-formed antibodies or antitoxins, which is there, like we have tetanus, snake venom, and all. It is required for quick immune response. So that is passive immunization. So that's it. Thank you.